The last speaker in this session is Alex Zhavoronkov. Alex is the founder and CEO of Insilico Medicine, a leading clinical stage biotechnology company, developing next generation generative artificial intelligence and robotic platforms for drug discovery. Since 2014, he has invented critical technologies in the field of generative artificial intelligence and reinforcement learning for the generation of novel molecular structures, the desired properties, and generation of synthetic biological and patient data. Under his leadership, Insilico raised over 400 million in multiple rounds from biotechnology, healthcare, and financial investors, opened R&D centers in 6 countries and regions, and partnered with multiple pharmaceutical, biotechnology, and academic institutions. The company has nominated 18 preclinical candidates so far, started 6 human clinical trials, and entered phase 2 with an AI-discovered novel target and AI-designed novel molecule. Alex, I can hand it over to you if you are ready with your presentation.
Ready with the presentation. Thank you, Shyam, and great to be presenting to you today. Thank you for this generous introduction and the ability to present with such esteemed speakers. Let me share my screen and start presenting. So my talk today will focus on the capabilities of end-to-end generative AI and robotics for drug discovery, and case studies showing what AI-powered drug discovery can do today and what we expect it to be able to do in the near future. There is a lot of hype around this area, and many companies are formed every day or every week. And of course, lots of progress in academia. Much of that is real, and I'd like to talk a little bit about the recent examples. So a little bit about Insilico. Just like Marisa presented Recursion, so we are also quite diverse and globally present. Our headquarters, global headquarters, is in Boston. We just moved it from New York, and so 1000 Mass Avenue. Welcome to join. Our AI is done in Montreal, Canada. That's where we do theoretical AI, applied AI, and theoretical AI is also done in Abu Dhabi. Beautiful place to be present in. Highly recommend everybody visiting. And we also have presence in Greater China, small team in Hong Kong, Taipei, Shanghai, and Suzhou. Shanghai, a pretty sizable team. And we utilize generative artificial intelligence to very rapidly design and discover drugs. However, our mission as a company is to extend healthy productive longevity for everyone, and our grand goal is to solve aging. This is how the company is organized. So a pretty important slide. We have our own LLMs that help orchestrate domain-specific models. Of course, we also utilize large foundational models to create some of the agents that we use everywhere. We have over 800 domain-specific models. Some of them are transformer-based, some are GANs, variational autoencoders, some are diffusion transformers. And what LLMs are really good at is orchestrating. So if you have API to those models and great description, you can have agents even creating research plans for you using domain-specific models. And currently we have over 2,000 agents in the company that we've created. So we pretty much replaced a lot of kind of low-level data annotation and even some of the coding tasks with agents. We have software in the market. Currently, 10 out of the top 20 pharma companies are using our tools. We protect ourselves from their data. However, we really like to receive feedback on the models, and that feedback is invaluable. It goes back into our QA and QC programs, and we do reinforcement learning with expert feedback. So we regularly retrain the models and make them available for the community. And we use those same models to put the drugs in clinical trials ourselves. We work with small molecules, biologics, and we also do target discovery. We also do a lot of work on aging research, so please follow us on Google Scholar. We usually put out maybe one or two interesting papers in aging research every quarter. And we started also utilizing our generative chemistry capabilities for agricultural tasks, so pesticides, herbicides, and also for CO2 capture material design tasks. So looking for molecules that bind CO2 efficiently. We get reasonably good revenue from that as well. So it's not just effective altruism, it's really interesting business. And now I'm going to show you the most important slide during this for this talk. Very often, people try to project what drug discovery is going to be with AI in the future in terms of speed, cost, and probability of success. However, there are very few benchmarks out there that can be used to see, okay, well, are you outperforming or not? So here are some internal benchmarks from Insilico. Many of those are date stamped with press releases or academic papers. So in 2019, we raised our first big round to do our own drug discovery and also put the software in the market. And since then, we've nominated 20 preclinical candidates. 9 reached IND clearance. 2 are in phase 2. 1 just completed phase 2A with safety and dose-dependent efficacy. And across all these, now our average time to preclinical candidate is 13 months. Shortest time to preclinical candidate was 9 months. That was QPCTL target. That one was actually also partnered. So it has already progressed into phase 1. But from 0 to preclinical candidate with a novel target, QPCTL is a novel target for cancer, we managed to achieve preclinical candidate in 9 months. For us, a PCC package usually includes at least 2 animal efficacy models and 28-day non-GLP talks. So it's pretty comprehensive and at minimum 2 models. In our TNIC paper, we've demonstrated that we've put out 18 experiments in one paper, preclinical, and that took us 18 months. That is our longest program for TNIC. 2022, we nominated 9 preclinical candidates. I think that if we go at current capacity and had unlimited funding, we could probably do 12 preclinical candidates. And we currently do not have any shortage of promising targets. I have more than 400 targets in stock to go after if anybody wants. And usually we try to balance target and compound novelty, confidence, and commercial tractability. So those are the three key criteria when selecting the program, because at the end of the day, you also want to ensure that the program is marketable. Our job is not to take every program into the clinic ourselves. We try to develop the drug to the point where it's de-risked, and then big pharmaceutical companies take it over and do later stage clinical trials. So how do we look at novelty? We usually look at whether the target has been already in the clinical trials. Is there clinical POC, other selective compounds, how many publications? So for example, for TNIC, when we started working on that program, I'm going to talk about it today. There were only, I think, 70 papers just mentioning the gene in different contexts, no selective compounds, no clinical POC, no clinical trials. So that was pretty novel, and we usually consider that to be maybe 75% novelty. And the compound novelty, usually do Tanimoto scoring with everything that has been developed against the target. discovered against this target, and then we have to look at, is it going to be first in class, best in class, patentable, differentiated? What is the patent life? So for our TNIC program, it was an absolutely novel scaffold, less than 60% to similarity with everything known. This is the slide that shows how Insilico operates. So we have software for science. You can buy most of those commercially. We have 4 verticals: biology 42, chemistry 42, medicine 42, and science 42. So biology 42 allows you to discover targets utilizing more than 60 different target discovery philosophies. So 23 different models in one place. So depending on your kind of head of the therapeutic area, what he or she likes, Pandomics will be able to satisfy the requirements most likely. If not, if they have their own philosophy, you can tell us about that philosophy. We will integrate it into Pandomics if it has reasonably solid theoretical base. We have a system called Generative Biologics that allows you to generate antibodies, peptides, and nanobodies. Currently, we started providing this platform either in pilot mode or some customers started already buying. We don't have our own biologics programs yet. And some of the software is open source. Some of the very exciting programs, please have a look, are live models. So think of like OpenAI's Sora that can generate 1-minute high quality video. We do similar things with biological data and Precious 3 GPT, multi-omics transformer, multimodal, multi-omics, multi-species transformer is currently open source. And you can play with it on Discord. So we're building a community to understand biology in time. Chemistry 42 allows you to generate molecules with the desired properties. Currently, it has over 40 generative models and over 700 predictive models for reinforcement learning. Very stable system. Alchemistry allows you to do DFT very cheaply and accurately. It comes free with Chemistry 42. Medicine 42 allows you to predict outcomes of clinical trials. Have a look at some of our published papers where we did retrospective, prospective, and quasi-prospective validation. And Science 42 allows you to work with outside pharma. And also we have a fun tool called Dora. It allows you to spawn teams of AI researchers that go and perform different research tasks, then kind of bring the research data back, and it gets compiled into a research paper. It's $17 a month. You can get it from our website. And we use these tools ourselves, whatever we provide to the community, to validate them using our own programs. So the first program that we started in 2019 is TNIC. It just completed our phase 2A. With good safety and unexpected dose-dependent efficacy with actual improvements in FVC. So we're sitting on some good data. It also showed efficacy in kidney fibrosis and several other types of fibrosis, but here we nominated 2 preclinical candidates. And we balance the rest of the pipeline with different capabilities of our platform. Balancing novelty, confidence, and commercial tractability. Some of those programs we have already licensed. So USP1 was a pretty successful licensing exercise to Exelixis, $80 million cash upfront when it reached IND. So not a super novel target, but very high confidence one. And we've had best-in-class chemistry, very differentiated. Cat6 was out-licensed to Menarini. It's over $500 million deal, $12 million upfront at preclinical candidate. It has progressed really nicely, and many of these are under term sheets. So we are continuing to supply pharmaceutical companies with high-quality drugs. Usually, to be able to license out an AI-discovered therapeutic, you need to have very high level of quality of the data room. So that's why we went to multiple pharmaceutical companies and even biopharmaceutical companies in Japan to learn how to create data rooms that exceed the quality standards of large pharmaceutical companies and ensure that every check mark is checked. Usually the level of quality that you need to achieve for an external licensing deal is actually higher than big pharmaceutical companies have internally. So it's very, very important to commit yourself to ultra high level of quality. Also, many of our tools have research papers explaining or application notes explaining how they work. So for PandaOmics, you can read the application note, how it works, and it explains some case studies and how models work. For Chemistry 42, also in one of my favorite journals, GSCAM, we put out the application note explaining how it works. Now, several smaller companies actually also picked up this model and started developing and providing tools in a very similar model. So deep reinforcement learning, I highly recommend looking at this paper and using this approach because it's pretty validated. Dora is a super cool tool. You can actually get it for, again, $17 a month or with 50% discount. Previous speaker, Marissa from Recursion, was speaking about RBM 39. Why don't we actually write a research paper about that in a, like, while I'm presenting? So let's do RBM 39. And I think they went for lymphoma. You can add additional papers here yourself. So the objective of every scientist is usually to write a paper. So we allow you to write a paper here. So it's always in the form of a paper. You can add your own data here, but let's say we go autopilot. So give it a second, and it will provide you with a research plan. So now the system is providing research plan based on my request. So it will perform omics data analysis, go through thousands of data sets that have been integrated into PandaOmics, perform network analysis, molecular hypothesis development. But let's say we also want to provide, perform comprehensive literature review and submit, generate content, let it run. So now you can see that it spawned an army of agents. You master agents as a principal investigator, omics data analysis for methylation, transcriptomics, knowledge graphs, web researcher, science reference finder. And by the time I finish talking, we will have a paper with original bibliography. So let's go back to the presentation while we're writing. So I just wanted to cover a little bit of history of generative AI in drug discovery. So in 2016, there were 3 competing groups, as I remember it, in this field. So one was ours. But of course, a very famous group is Alana Spuruguzic's lab and Hongmin Chen's lab at AstraZeneca. So Alan published his first very, very famous paper with Gomez Bombarelli, automatic chemical design using data-driven continuous representation of molecules. It went on archive in 2016 in October. I had to tear some of my hair because mine was in peer review in an academic journal at the time. And similar idea, and it got published and now very highly cited on ICS Central Science. Hongmin Chen, also a very early works in 2017, molecular de novo design through deep reinforcement learning, and many others. We together published Moses. Some of my papers, so our first one was 2016, but somehow it shows 2017, cornucopia of meaningful leads. We used GANs for small molecule drug design, but didn't do any experiments there. And then really big experimental validation study for us for generative tensorial reinforcement learning was 2019. Here is a timeline of some of those experiments. So green is Insilico papers. Our first experimental study was entangled conditional autoencoder, where we designed our first selective JAK3 inhibitor and published and tested it. We did it in 2017, but we managed to publish only in 2018, and then 2019 was Generative. And the greatest bottleneck, we very quickly realized that it's experimental validation. It takes you sometimes, 5 weeks to create and train a new model, new generative model, regardless of how complex it is. Usually it doesn't take longer, but it can take you several years to ensure that all the validation experiments are confirmed. By the way, a great paper that appeared in Nature Machine Intelligence this April out of Tom Blundell's lab at Cambridge, when I haven't seen it before it got published, but when I read it, I was blown away how much work they performed. So they actually looked at all the validation experiments that appeared in literature for every model published for generative chemistry with input, output, design, task, target, hit rate, result, and year published for distribution learning and also for goal-oriented learning. And these are some of our experiments. So I was very happy to see that our work was very well represented. So we did publish quite a bit with experiments. And here are some case studies I would like to present. So do compounds designed using AI work experimentally? So that was our 2019 paper. We did the experiment 2018, took almost a year to publish, almost got rejected, and now pretty highly cited. So here we were challenged by our partner for any target out of the hat. They gave us DDR1 kinase, 21 days to generate 6 compounds. Then we synthesized and tested. We did it with Alana Spurogudzik. He was the last author. I was the first author. So at that time, we became friends during this experiment. Pretty famous paper. And in 46 days, we managed to dose the mouse. And out of those 6 compounds, 4 worked. 2 went through microsomal assays, and 1 was tested in mouse. We were actually limited by the time and also the ability to synthesize. So, of course, some of those molecules shared some structural similarity with known compounds. And we did not deny that, but the fact that generative tensorial reinforcement learning managed to do that very quickly was pretty impressive. Then it was criticized by some of the competing groups, and we responded and explained that it's scalable. But at that time, again, people were debating the potential of generative chemistry in this industry, and we had to respond. But then once we managed to raise some money, we started doing real programs, and you've seen some of the results, 20 PCCs since 2019, since that paper was published. And some of the POCs we've managed to publish without going into commercial programs. So one really cool experiment was this one. We did it in 2021, at the end of the 2021. So we used, at the beginning of 2022. And we used Pandomics to identify CDK20 as a novel target for hepatocellular carcinoma. Used AlphaFold protein structure and chemistry 42 to very rapidly do 2 rounds of synthesis. So in first round of synthesis, we got 7.3 micromolar hits, so not great. Gave it back to chemistry 42 for a ligand-based drug design and got 180 nanomolar hits, so not too bad, and tested in vitro, and they worked, and we published it in Chemical Science. So currently, one academic group is taking this a version of this molecule forward, which was very cool because they know HSC really well. And we also can use AI to repurpose existing drugs. Some companies actually developed business models around this philosophy. So in this particular experiment, I presented in March at NCLS Consortium 2022, and met brilliant Merit Chutkovits and Jeffrey Rothstein, who run NCLS. We applied, got the data, combined it with our Pandomics data set that we have in the database, and got several new targets and also drugs to repurpose, tested them in a fly model out of Mayo Clinic with Janke. And out of the 26 predicted targets, 18 showed strong or moderate rescue and in fly models, and only 2 showed moderate enhancement of the ALS phenotype. And Lu Bai, at that time dean of the medical school at Tsinghua University, now he is in Shanghai, very famous American Chinese scientist in neuroscience. He also is a co-founder of 4B Technologies. So they picked one of the targets that we've identified during that experiment, took also one of the repurposing candidates, and in under 1 year, they enrolled 64 patients in the clinical trial. So the readout for this one is expected before the end of this year, so fingers crossed. But that's also shows you the power of China. So some of the diseases are not that rare. And if you want to do a quick investigator-initiated study, you can actually do that. And I think that this is where the power of AI has to be explored and exploited. And then can we go after a novel target with a novel molecule for a real commercial program? And it's very difficult and rare. As a matter of fact, if you were to survey my Twitter followers, have China ever discovered a novel drug, novel target, novel molecule? The answer would be usually no. And some people debate, but those are usually older targets. If you ask ChatGPT Preview the same question, the answer would be usually also no. And this is the prerogative of very well-developed countries. So actually, if you look at also ask ChatGPT o1 Preview, how many countries ever discovered a novel drug, novel target, novel molecule, there are actually not that many. And for targeted therapeutics, which is actually pretty scary as well. So we need to get more players to the molecular casino. That's why we also released the software. And we wanted to try and go after this goal ourselves from the very beginning without repurposing, with novel target, novel molecule. We actually thought that we would fail. That's why we started recording a video documentary around this target and around this program, but we succeeded, which was very unexpected. This program is described in this paper that was published in Nature Biotech 8th of March 2024, pretty popular paper. In this paper, we described how we identified the target, did hallmarks of aging assessment. That's kind of a long story. I'm not going to go too deep into that because I'm probably a little bit out of time. Synthesized only 79 molecules for this program. By the way, our average now per program out of over 20 preclinical candidates, we synthesized around 70 molecules per program. So if you are to benchmark, that's around that number. So here we did 79. 055 is 55th molecule that we synthesized. We did indication prioritization. TNIC, we believe, should work in many indications, but IPF was the highest probability and also most commercially tractable route. Performed multiple in vitro experiments, multiple in vivo experiments, nominated 3 preclinical candidates in 1 paper. Did indication expansion, phase 0 in Australia in 8 healthy volunteers, 2 phase 1s in New Zealand and in China. And now, by the way, if you want to talk to this paper, since it has lots of supplementary materials, we also created a conversational interface. In June 2023, it was actually very cool. Nowadays, you can even do it with ChatGPT. And I'll skip this. But now we have completed the China phase 2A. And observed very good safety and tolerability. So primary objective was met, but we also met the secondary objective. So we showed dose-dependent increase in forced vital capacity, which was very unexpected. Currently, I'm not going to show you the data. We're working on a high-profile academic publication. So if you know anybody in high-profile clinical journal, ping me, please. we're submitting, but now this is the program that is likely to increase the confidence in the entire AI drug discovery industry. I'm going to skip a few things. By the way, if you like to see how an ultra-modern robotics lab works, please welcome to our lab in Suzhou. We have 6 interconnected rooms, fully automated, that work with cells, tissues, organoids. They can do sample processing, and for every sample, we usually get methylation, transcriptomics, whole genome, and 4 or 5 types of imaging. Then AI processes this data, picks the targets, and for the targets that we might have tool compounds, picks tool compounds, incubates the original sample with those tool compounds, and performs the entire sequence one more time to see if the predictions were confirmed. If they were, and there are some targets that don't have tool compounds and score higher, we perform CRISPR and sRNA studies. And for those that scored high and were unexpected and have tool compounds, we actually prosecute them further as well. And we now have a very large database of targets. So currently, if you need targets for multiple types of cancer, for fibrosis, muscle wasting, IBD, please welcome to talk to us. And now I'm out of time, I know.
Are you in your final slides?
Yeah, sure. So I just wanted to mention that aging research is also very, very important for drug discovery. Many biological processes happen in time, and we go through our life from cradle to the grave. Deep neural networks are very powerful tools to actually capture the patterns from cradle to the grave, even without disease or with disease. And we like to discuss the capabilities of aging research as a platform for drug discovery at the conference that we co-organized. Now it's going to be the 12th year in Copenhagen. It's a 5-day event. Please welcome. Usually it gets sold out at least 2 months in advance. So big pharma, academia all come together. Please join, and happy to host you there.
Yeah, thank you so much, Alex. That was quite impressive. Based on the comments, it seems like the work that you are doing at Insilico is an inspiration for many of the folks. So I think looks like that is the new standard, probably what you're setting over here. But we'll just take a couple of questions. One of the questions was, how much computing resources can I expect to spend in a virtual screening in your tools? And the second, related to that, is you mentioned that there are no good benchmarks for hit ID models. CASF and CSAR are widely used. Are those not applicable for you?
So sure. Well, first of all, most of what we do are generative modeling. So not only virtual screening. Virtual screening, actually, in our case, reinforcement learning is very cheap. So usually for one exercise to get to a hit for kind of a reasonably difficult target, you would spend about 72 hours in compute and maybe about $5,000 in addition to annual license for the tool. That would involve generation and screening at the same time, because you generate using 42 models and then you screen using more than 700 models. We also have alchemistry, which allows you to do physics-based modeling, but that one is super cheap as well. Usually when you're left with like 20 molecules out of chemistry 42 and you want to get higher confidence, you can do DFT, docking, and that would cost you an additional maybe $1,000. And traditional benchmarks for hit ID models, of course, they are applicable. But we have our own pipeline for hit ID, so we already established our own benchmarks.
We will take one more, probably last question, and if you can stay online to answer some of the other questions, that will be great. One of the questions over here is, in case of an absent crystal structure, you have shown one success. Have you tried other targets while going with the same pipeline? If yes, how many of those resulted in a success?
Yeah, if you're talking about AlphaFold, we also published one on SIG2 target POC study. Very rarely we would go for targets without crystal structure for a commercial program. Usually for a commercial program, when you're actually going at, if you're planning to invest, $20 million in a program, you probably get 4 or 5 crystal structures because it's, $30,000 and 2 weeks or 3 weeks, depending on which CRO you use. So my recommendation is that always get a crystal.
I would like to thank all the speakers. Thanks, Alex, again. I would like to thank all the speakers in this particular session, Sean, Woody, Marisa, as well as Alex.